Imagine a battlefield hidden inside your own body, where rogue cells evolve faster than any army and the immune system both fights and forgets its own war. In the world of cancer biology, the first surprise is that tumors can literally masquerade as normal tissue by hijacking the body's DNA, becoming invisible to the vigilant T cells that normally patrol our veins. This stealth mode lets cancer grow unchecked, a secret weapon that scientists only uncovered in the last decade. Next, discover that our own defenses can be turned off. Tumors unleash molecular brakes such as PD-L1, which snuff out attacking T cells and let the disease spread. Yet hope rises with CRT therapy, where doctors reprogram a patient's T cells into smart missiles that lock onto cancer cells with razor-sharp precision, delivering a one-way strike that ordinary immunity can't achieve. The challenge doesn't end there. Cancers mutate at breakneck speed, constantly reshaping their surface proteins, which means vaccines often chase a moving target and fall behind. Finally, checkpoint inhibitors have turned terminal diagnoses into survivable stories, but they work for only about 1 in 5 patients, exposing the tangled complexity of tumor-immune interactions. These 5 facts reveal a hidden world of genetic camouflage, immune sabotage, engineered warriors, relentless evolution, and uneven breakthroughs. But that's just the beginning. In Part 2, we go deeper. Welcome to Part 2 of our deep dive into the biology of cancer and immunotherapy. In this episode, we travel from the earliest whispers of immune involvement in cancer to the cutting-edge mechanisms that power today's life-saving treatments. In the 19th century, Rudolf Virchow's pioneering work suggested that tumors behave like unhealed wounds, a concept that lay dormant for decades. It wasn't until William Coley's daring toxin experiments, injecting bacterial extracts into sarcoma patients, that the idea of harnessing the immune system began to surface. Fast forward to 1971, when researchers isolated tumor-specific antigens, confirming that cancer cells could indeed display unique flags for immune recognition. This breakthrough paved the way for interleukin-2, the first cytokine approved by the FDA in 1992, which demonstrated that immune activation could shrink metastatic melanoma. The scientific landscape transformed dramatically with the discovery of immune checkpoints. In 2010, James Allison identified CTLA-4 as a brake on T-cell activity, while Tasuku Honjo uncovered PD-1's role in suppressing immune responses. Their work, honored with a Nobel Prize in 2018, ushered in a new era where antibodies could lift these brakes, empowering the body's own defenses to target malignancies. Understanding the molecular dance of DNA mutations such as KRAS oncogene activation and TP53 loss reveals why some tumors are more immunogenic than others. The tumor microenvironment, densely packed with fibroblasts, macrophages, and suppressive cytokines, creates a hostile arena for T cells. Tumors exploit PD-L1 expression, secrete indolamine 2,3-dioxygenase, and downregulate antigen-presenting machinery, effectively cloaking themselves from immune surveillance. Modern immunotherapies exploit these insights with remarkable precision. Checkpoint inhibitors like pembrolizumab and nivolumab bind PD-1, preventing tumor-derived PD-L1 from silencing T cells, and have shown durable responses in lung, skin, and kidney cancers. CRT therapy takes a different route, extracting a patient's T cells, genetically reprogramming them to express a chimeric antigen receptor targeting CD19, and reinfusing them to eradicate B-cell leukemias. The results, complete remission in heavily pretreated patients, highlight the power of engineered immunity. Cancer vaccines, once a failed concept, have resurged with neoantigen-focused designs that tailor peptide cocktails to a patient's unique tumor mutational landscape, prompting a bespoke T-cell attack. Oncolytic viruses, such as talimogene laherparepvec, simultaneously lyse tumor cells and amplify local immune activation, turning cold tumors hot. Expert voices stress that monotherapy rarely succeeds against the adaptive capabilities of cancer. Combination regimens, pairing checkpoint blockade with radiation, targeted kinase inhibitors, or personalized vaccines, are now the frontier of research, aiming to overcome resistance pathways. Biomarker development, including tumor mutational burden and microsatellite instability, guides clinicians in selecting patients most likely to benefit from immune-based strategies. As we stand at the intersection of molecular biology, genetics, and immunology, the promise of curative approaches grows ever brighter, but the complexity demands continuous learning and nuanced application. In Part 3, we uncover what mainstream sources won't tell you. Imagine a battlefield you cannot see, hidden deep within your own body. Every day, millions of cells wage a silent war, but what you think you know about this conflict is only the tip of the iceberg. In the first act, we discover that tumors are not isolated islands. Instead, they sit atop a shadowy arena where rogue cancer cells and vigilant immune soldiers constantly clash. This hidden confrontation reshapes the very foundation of our understanding, turning a simple disease narrative into a complex tale of espionage. Just when you think the immune system is an unwavering guardian, a twist emerges. Some of its own checkpoints become double agents. These covert allies allow malignant cells to slip past detection, masquerading as normal tissue and deceiving our defenses. The revelation that the body's protectors can betray us flips the script on every conventional treatment we've trusted for decades. Then, a breakthrough arrives like a secret weapon: immunotherapy. By unlocking the true potential of the immune system, scientists can reprogram those double agents, forcing tumors to expose themselves. This radical shift doesn't just improve survival rates, it rewrites the entire playbook of cancer care. Yet, the precise mechanisms behind this alchemy remain tightly guarded, a mystery that fuels both hope and intrigue. As we peel back each layer, the suspense deepens. What hidden signals are we still missing? How many more covert allies lie dormant, waiting for the right key? The answers lie just beyond our current reach, urging us to keep watching. Now that you know the truth, Part 4 will change how you see this forever. Welcome to the finale of our 4-part series, where we unlock the deepest layers of cancer biology and immunotherapy. We start by traveling back to the earliest records of malignant disease, noting how ancient physicians described hard, invasive growths, but it was not until Rudolf Virchow coined the term carcinoma that pathology began to reflect cellular dysfunction. Fast forward to the late 1800s, William Coley's daring use of bacterial extracts hinted at the immune system's capacity to recognize and attack tumors, a notion dismissed for decades until the molecular era revived it. In the 20th century, breakthroughs in immunology revealed that tumors are not invisible to the immune surveillance network. Instead, they actively sabotage it through a suite of evasion tactics. Tumor cells overexpress PD-L1, secrete TGF-beta, and recruit myeloid-derived suppressor cells, creating an immunosuppressive niche that thwarts cytotoxic T lymphocytes. This understanding catalyzed the development of checkpoint inhibitors, antibodies that free the brakes on immune activation. The landmark 2011 approval of ipilimumab for melanoma heralded a paradigm shift, and subsequent agents targeting PD-1 and PD-L1 extended survival across lung, kidney, and bladder cancers. Parallel to checkpoint blockade, chimeric antigen receptor T-cell therapy reprogrammed patient-derived lymphocytes with synthetic receptors, enabling them to seek out and destroy malignant B-cell clones with unprecedented precision. Clinical data now report remission rates exceeding 90% in certain pediatric leukemias, confirming that engineered immunity can outmaneuver even the most aggressive malignancies. Yet the journey is far from complete. Tumor heterogeneity, loss of target antigen expression, and adaptive resistance continue to challenge clinicians. Current research explores bispecific antibodies that bridge T cells and tumor antigens, personalized neoantigen vaccines derived from individual tumor sequencing, and oncolytic viruses engineered to selectively lyse cancer cells while priming systemic immunity. The microbiome emerges as a hidden influencer, modulating response to checkpoint inhibitors through metabolite signaling pathways. As these modalities converge, the field faces profound ethical and practical dilemmas. The cost of CAR T therapies, often exceeding $500,000 per patient, strains healthcare systems and raises questions of equitable access. Immune-related adverse events, ranging from colitis to myocarditis, demand multidisciplinary management and highlight the fine line between therapeutic vigor and collateral damage. Moreover, the prospect of editing germline immune cells invites societal scrutiny about long-term consequences. Despite these challenges, the momentum is undeniable. Ongoing trials combining checkpoint blockade with radiation, targeted kinase inhibitors, and metabolic modulators aim to rewrite the rulebook of cancer treatment. By integrating genomic profiling, real-time immune monitoring, and artificial intelligence, the next decade promises a truly personalized oncology landscape where every tumor's unique fingerprint guides a bespoke immunologic attack. In closing, remember that the story of cancer and immunotherapy is a testament to human ingenuity, a relentless pursuit to turn a fatal disease into a chronic, controllable condition. If this series gave you value, subscribe, more deep dives coming every week.